Good day, everyone. I hope you're all ready to learn another topic this day. So, for today, we're going to discuss the fifth laboratory exercise, which is about the production of polyhydroxyalkanoates from soil bacteria. Specifically, this report is from a journal article entitled The Metabolic Relationship Between Polyhydroxyalkanoic Acid and Rhamnolipid Synthesis in Pseudomonas aeruginosa, a comparative C-NMR analysis of the products in wild type and mutants. I am Angelica Cabantac, and with me are Miss Buhayan, Miss Kaba, and Miss Kabugnason.
Polyhydroxyalkanoates or PHAs are polyesters produced in nature by numerous microorganisms, including through bacterial fermentation of sugars or lipids. When produced by bacteria, they serve as both a source of energy and as a carbon store. Bacteria can synthesize a wide range of biopolymers that can serve diverse biological functions with material properties suitable for numerous industrial and medical applications. Various bacterial species accumulate intracellular polyhydroxyalkanoates granules, which are biodegradable, environmentally friendly, and biocompatible thermoplastics. pH have been studied extensively as biomaterials for the replacement of petrochemical-based plastics. They can be either thermoplastic or elastomeric materials. In addition, PHAs still have only a very limited market because of the availability of large amount of shale gas and petroleum that will not rise dramatically in price, which makes PHA less competitive compared with low-cost petroleum-based plastics. Therefore, strategies must be adapted to meet this challenge by developing a super PHA production strain combined with advanced fermentation processes to produce PHA at a low cost, and by the construction of functional PHA production strains with technology to control the precise structures of PHA molecules allow the resulting PHA with high value-added applications. At the end of the exercise, the student should be able to achieve the following goals as measured in the learning outcomes. First, isolate and screen polyhydroxyalkanoic acids producing bacteria. Second, optimize the production of biopolymer PHAs under various growth conditions. And lastly, to find direct evidence showing a competitive relationship between both pathways toward hydroxy acids.
Good day, everyone. So this is how the experiment was done. For bacterial strains and culture media, the bacterial strains used in this study are listed in the table, and cells of the Pseudomonas aeruginosa were grown in nutrient-rich medium with 1% yeast extract, 1.5 nutrient broth, and 0.2 ammonium sulfate for seeding, maintenance, and storage of bacterial strains. For PHA and rhamnolipid synthesis medium, the modified M1 mineral salt medium of the same composition as that reported earlier was used, and antibiotics were added at the following concentrations: the 60 microgram per ml tetracycline and 30 microgram per ml of gentamicin. For the monitoring of cell growth, the microbial strains was inoculated using a 5 ml of nutrient-rich at 30 degrees Celsius with 180 rpm for 12 hours, and it was transferred to 500 ml of M1 medium supplemented with 70 mm fructose or 30 mm decanoic acid and 1 gram per liter of ammonium sulfate in a 2 liters flask. The culture was aerobically cultivated until a steady state growth was reached. So the cell was determined by measuring the dry cell weight, or DCW. The cells were isolated by centrifugation and then washed with methanol and dried under a vacuum at room temperature for 24 hours. The concentration of ammonium ion remaining in the media was measured using Nessler's reagent method, and The concentration of fructose remaining in the medium was determined using the 3,5-dinitrosalicylic acid or DNS method. And to measure the concentration of decanoic acid remaining in the medium, cell-free supernatant was extracted with chloroform and then analyzed by gas chromatography. So this is the method for quantitative assay of PHA in cells. For the analysis of PHA in cells, 20 mg of dried cells were reacted with a mixture containing 1 ml of chloroform, 0.85 ml of methanol, and 0.15 ml of concentrated sulfuric acid. The reaction mixture in a closed screw-cape tube was incubated at 100 degrees Celsius for 3 hours, and the organic layer containing the reaction products was separated, dried over magnesium sulfate, and analyzed by gas chromatography. The sample was then injected under splitless mode, and each peak was standardized again against standard 3-hydroxy methyl esters, which were obtained by methanolysis of purified PHA with known compositions. And then it was determined by quantitative NMR analysis, and the column was heated at 8 degrees Celsius per minute for 80 to 250 degrees Celsius, and the temperature of injector and detector was 230 and 280 degrees Celsius respectively. In rhamnolipid assay, the orsinol assay was used to assess the amount of rhamnose in rhamnolipids secreted into the medium during cultivation on the modified M1 mineral salt medium. So a 300 degrees Celsius sample of the culture supernatant was extracted twice with 600 degrees Celsius diethyl ether. The ether fractions were pulled and evaporated to dryness. After heating for 30 minutes at 80 degrees Celsius, the samples were cooled for 15 minutes at room temperature, and the absorbance was measured at 421 nanometer. The concentrations of rhamnose in rhamnolipids were determined by comparing the data with those obtained with rhamnose standards between 0 and 50 mg per ml. Then the rhamnolipids content was calculated based on the ratio of the mono and dirhamnolipids depending on the type of strain and carbon source used, which was determined by comparing the peak areas of the 13C NMR signals characteristic to them. The acyl group component of rhamnolipids was analyzed by gas chromatography method, and culture supernatant was extracted with chloroform overnight after acidification to pH 2. The filtered chloroform extract was concentrated using a rotary evaporator, and an aliquot of chloroform extract was subjected to methanolysis reaction, followed by gas chromatographic analysis as described above. Lastly, here is the method in isolating the PHA and the rhamnolipid. To investigate the metabolic pathway of PHA and rhamnolipid synthesis from octanoic acid, PA14 wild type, PA14 Faz mutant, and PA14 Fag mutant were cultivated using 99% isotope C13 enriched octanoic acid at the position of C1. PHA was extracted from dried cells with hot chloroform in a pyroxolate apparatus for 8 hours. Concentrated solvent was precipitated in rapidly stirred cold methanol, and then the isolated PHA was purified by reprecipitating and dried overnight under the vacuum at 50 degrees Celsius. Rhamnolipid was isolated from cell-free culture supernatant by chloroform extraction, and the collected culture supernatant was first acidified to pH 2 with 6N hydrochloric and extracted with chloroform overnight at room temperature. The organic phase was then collected and concentrated by evaporation and allowed to yield the viscous rhamnolipid product. The isolated PHA and rhamnolipid were analyzed by NMR spectroscopy.
For the result and discussion, it was found that polyhydroxyalkanoic acid And rhamnolipid synthesis in P. aeruginosa have a close metabolic relationship since R3-hydroxy fatty acids are used as common precursors in both pathways. Therefore, it was assumed that both biosynthesis pathways may compete for R3-hydroxy fatty acid ACP. Rhamnolipid negative P. aeruginosa mutants were found to increase PHA accumulation compared to the wild-type strains, suggesting that in the rhamnolipid synthesis, it can block mutants. So R3-hydroxy fatty acids become more available for the PHA or for the polyhydroxyalkanoic acid synthesis. An interrelationship between the PHA and rhamnolipid synthesis was found that in rhamnolipid negative mutants, more R3-hydroxy fatty acid precursors were flux toward PHA synthesis. Let's proceed to the genes involved. There are four PHA mutant strains defective in genes involved in PHA accumulation, which includes PHA synthase genes, which are PHAC1 and PHAC2. We have PHA depolymerase gene, which is PHAZ. And lastly, we have trans-acylase gene, which is PHAG. P. aeruginosa, PA14, and PaO1, PHAC1 mutants grown on fructose showed a slightly decreased PHA accumulation, whereas when grown in decanoate, it was strongly impaired as compared to that of wild type. In contrast to the case of PHAC1 mutants, PHAC2 mutants were strongly impaired in PHA accumulation when grown on both fructose and decanoate. So, for the PA14 PHAZ mutant accumulated almost 300% more PHA than PA14 wild-type strain when cells were grown on a decanoic acid. The two PHAG mutants grown on fructose significantly decreased the accumulation of PHA and slightly decreased the secretion of rhamnolipid compared to the wild type, while when grown on decanoic acid, it synthesized almost the same amounts of PHA and rhamnolipids. Other genes involved are the RPOS and RH1A. RPOS mutant strains of PA14 and PaO1 was grown on 70 fructose, and the production of PHA is decreased to less than 10% and 40% compared to that of the wild type. Whereas in RHLA mutant, which lacks rhamnolipid production, it has an increased production of PHA.
Once again, good morning everyone. I am Shane Mariel J. Caba, and in this part of our report, I'm going to discuss the metabolic pathway and genes involved in the PHA production. Being economically important, it is necessary to study and understand the metabolic routes through which microbial PHA are synthesized. So, in definition, PHAs are biopolyesters synthesized by various microbes as a reservoir of carbon and energy in granular form. PHA synthesis advanced through three major pathways catalyzed by different enzymes and are connected to various biochemical pathways like glycolysis, the TCA cycle, fatty acid, and amino acid metabolic pathways. In this figure, you can see the schematic representation of the polyhydroxyalkanoates production through pathway 1, 2, and 3. Pathway 1 deals with the production of monomers of 3-hydroxybutyryl coenzyme A from acetyl coenzyme A molecules by the catalytic activity of the beta-ketothiolase and acetoacetyl coenzyme A reductase. These monomers are further polymerized to form polyhydroxybutyrate or the PHB. The acetyl coenzyme A molecules utilized in pathway 1 are formed by the metabolism of carbon sources such as sugar, fatty acid or amino acid. Glucose is the preferred carbon source in pathway 1. And in pathway 1, when glucose is used as the carbon source, pyruvate formed from glycolysis gets converted to lactate by lactate dehydrogenase. The propionyl coenzyme A transferase enzyme further converts this lactate to lactyl coenzyme A, which is further polymerized to form PHA. The succinyl coenzyme A from the TCA cycle is also directed to form the monomers of the PHA synthesis. The intermediary products of the fatty acid oxidation are utilized for the production of monomers for PHA synthesis. This pathway of the PHA synthesis, which uses fatty acids solely as its carbon source, is designated as pathway 2. Pathway 2 specifically synthesizes PHAs like hydroxyhexanoate, hydroxyheptanoate, hydroxyoctanoate, and hydroxydecanoate. And this pathway is predominantly active in microbes such as Pseudomonas aeruginosa. In pathway 3, R-hydroxyacyl-CoA monomers were formed from the malonyl CoA or the coenzyme A. The acetyl coenzyme A from glucose metabolism is converted to malonyl CoA by the acetyl CoA carboxylase. The malonyl coenzyme A then gets converted to malonyl CoA ACP by malonyl CoA ACP translase. The malonyl CoA ACP is converted to acetoacyl ACP and acetoacyl CoA by the 3-ketoacyl ACP synthase 3. The 3-ketoacyl ACP reductase catalyzes the conversion of the acetoacetyl CoA to 3-hydroxyacyl CoA, and these monomers are polymerized by PHAC enzyme to form PHAs. So the outlines of all the pathways from pathway 1, 2, and 3 is what is shown in this figure. Next is how are we going to optimize the processes or the technology involved in the production of PHAs, particularly the polyhydroxybutyrate. So in this figure, this is a representation of workflow in bioprocess technology for the production of the PHAs using cyanobacteria as its host system. So just a little definition of cyanobacteria. They are gram-negative bacteria with a long evolutionary history and are the only prokaryotes capable of plant-like oxygenic photosynthesis. So unlike heterotrophic organisms, cyanobacteria require only greenhouse gases like carbon dioxide and sunlight along with minimal nutrients for growth, eliminating the cost of carbon source and complex media components. So the cyanobacteria are equipped with superior photosynthetic machinery showing higher biomass production rates compared to plants and can convert up to 3 to 9% of the solar energy into biomass. Some cyanobacteria can produce PHB when essential nutrients for growth such as nitrogen and phosphorus is limiting. From an economic point of view, however, photosynthetic PHB production in cyanobacteria has two major disadvantages. So the first is little productivity and second is slow growth. So therefore, in order to promote photosynthetic PHB production on an industrially relevant scale, the productivity needs to improve significantly. So the figure you see here represents a bioprocess development chain for cyanobacterial PHA production consisting of strain and bioprocess developments and the downstream processing. The workflow shows the strain selection, the strain improvement, the process understanding, and the strategies for scale up, and then also the downstream processing, representing the separation and purification of the final product. So, in order to obtain an optimal scalable bioprocess and in-depth understanding of the bioprocess kinetics, the following points have to be known. So, first, the holistic knowledge of enzymatic and metabolic pathways of the PHB biosynthesis in cyanobacteria. Second is the selection or the development of the optimal strain with maximum productivity. Third is the selection of inexpensive substrate and optimization of media components for the particular production strain. Fourth is the design of the bioreactor system and the optimization of process parameters for scale up using the statistical design of experiments. Fifth is the usage of past data and advanced mathematical models for monitoring and control. And lastly, the development of these novel strategies for PHB recovery with a minimum cost of energy and chemical requirement.